This class is a potpourri of learning, including some practical applications based on the principles of learning, which makes about a panoply of very useful programs and therapies. What we will look at is a tiny bit of comparative cognition on a topic called the episodic-like memory. This is a sophisticated memory skill of putting together what, where, and when information together. We will look at a case of social learning. So social learning is a whole mix of learning, including imitation, but also a lot of other means of social learning. You will see what mechanism is at work when we come to the case. The case is very interesting because in this case, the learned practice is transmitted from generation to generation. It makes it a form of cultural transmission, and it's found in black rats. And then we'll look at 3 practical applications. One is training the brain. What that means is positive reinforcement for particular kinds of brain activity. You don't have to do anything other than think to get reinforcement in these conditions. And then we'll look at 2 lines of behavior therapies for treating phobias and to stop disastrous behaviors such as kids slamming their heads into walls or babies vomiting foods constantly. We will start with a story in comparative cognition on episodic-like memory. And this is a very influential story. It's got quite a splash in the comparative cognition literature. Episodic memory for humans, this is a term that's coined by psychologist Endel Tulving, one of the most prolific and influential psychologists of all time. He's a memory psychologist. So what Tulving means by episodic memory is actually remembering an episode. And this generally comes with the notion that you know when the event takes place, you know where the event took place, and you can also visualize, right, what event has taken place. And that's the autonoetic consciousness, okay? So what it means, sort of this autonoetic consciousness, it's a complicated term that Tulving made up, is that you could actually see yourself having breakfast, right? If you think back to breakfast this morning, can you picture yourself there? Yeah, that's autonoetic consciousness, okay? Now we can't ask that. You can't ask an animal other than a human whether you can picture yourself doing, at this event X, but we can ask about what, where, and when components. So in that case, that's why this term like is added there. So episodic like memory means what, where, and when memory. We don't know about autonoetic consciousness, can't ask that about any other animal. The case we want to focus on comes from Professor Nicola Clayton's lab, Nikki to her pals. In 2009, Cambridge University celebrated its 800th birthday, whoa, with a lot of fanfare. So Clayton, among other things, not only a bird psychologist, but a keen ballroom dancer, produced a clip called Bird Tango, right, in which she mixes dancing in her science with birds. Pure infotainment, very entertaining. The research here was done with another psychologist at Cambridge, Tony Dickinson, in the Department of Psychology. Scrub jays are the subjects, and they are food-storing birds, right? They're in the same family as the Clark's nutcrackers that we met in earlier in one of the weeks on evolution. Scrub jays, however, don't store as prolifically as Clark's nutcrackers. But they are willing to store and retrieve foods in the lab. For example, on these ice cube trays, right, filled with sand. And these different ice cube trays, they're marked with these Lego pieces. The Lego pieces, they are serving as landmarks. So they will bury foods and they will try to find the food again. Here's the key experiment. So these birds eat a variety of insect and plant foods, but they love worms. They love mealworms, or in this case, waxworms were used as a favorite food. Waxworms are rich in fat and protein, and birds generally love them. So in some pre-training, the birds learn that the stored waxworms will rot over 5 days, whereas peanuts, a less favored food, are okay even after that delay. So experimentally, what the experimenters did was degraded the worms to make sure of these contingencies. Basically, you can add something yucky tasting to the worms so that they're awful after 5 days. Okay, so after 1 day, or after less than 1 day, the worms are okay. After 5 days, the worms degrade. Birds were just given a couple of trials of this kind of pre-training, and then comes a key manipulation. And that's to let the birds store foods in ice cube trays on 2 different occasions. On each occasion, only one food was available for storing, and only one side of the tray was open. The other side was covered with perspex, so the bird can't store food there. So these 2 caching experiences were separated by 5 days. All right? 4 hours after the second caching session, they were allowed to retrieve the food. All right? So that's caching experience 1, caching experience 2. 4 hours later, there's a retrieval test. So, if they catch the worms last, as in this condition, and then they've cached it 4 hours earlier, you would predict that the birds who like the worms would choose the worm first, choose the side where they store the worm first. Okay. On the other hand, if they store the worms 5 days ago, and then they store the peanuts just 4 hours ago, you would predict that they would think, well, the worms are now not so good, and under this condition, they should choose the peanut first. Okay, those are the predictions for this case of if they have what, where, and when memory. They can put events, times, place, and the food all together into the memory. And that's exactly the pattern found on the test with all the food removed. So on the test, there's no food there. You just test where the birds go to search. A nice touch here with this, the scientists actually scoring the birds did not know whether what they had stored or what condition the worms were in. So the behavior was scored blind, so they couldn't. They try to wipe out experimental bias from the scoring system. Very nice touch. No food on the test means that there are no cues from the food. Okay, the birds must be relying on memory. They can't possibly smell anything there because there are no food there. So the top panel shows the key condition. If the worm was stored 4 hours ago, that's under this column, they overwhelmingly prefer to search for the worm first, right? And they search most for the worm. If the worm was stored 124 hours ago, however, then they don't show much interest in searching for the worm. They show far more interest searching for the peanut. Okay? This bottom panel here is just the control condition. It's far less interesting. It's in the condition in which the birds have learned that worms do not degrade, right? Even after 5 days. And then the worms do not degrade even if they store the worm long time ago, they still prefer to go after the worms. Okay? So to do this, you've got to put what, where, and when information together. You got to know when the food was stored. You got to know where you've stored the food. You got to know what food you've stored, right? So to perform well on this particular task requires putting together what, when, and where information. And that's what makes it a case of episodic-like memory. I should say that a similar pattern can also be found in rats, in an experiment using quite a different design that I won't tell you about. So, while we're not sure about autonoetic consciousness in any of these animals, one bird and at least one non-human mammal has been shown to possess something like episodic-like memory. Story number 2, social learning in these black rats. This is a story in which learning provides a means of transmitting a way of life. So this is a way of life that's transmitted down generations in a particular population, these black rats. The story takes place in Israel, and it begins when high school biology teacher Ran Asner discovered pine cones in stands of Jerusalem pine that had been stripped of their seeds. So all these pine cones on the ground have been stripped of their seeds. So Israel had planted many of these stands of Jerusalem pine, right, throughout the latter half of the 20th century. This here was obviously the work of some animal, but what was it, right? Well, you have to trap the rats. You have to trap the animals to try to find out. And that took a long time because they were setting traps on the ground, and actually the animals weren't on the ground at all. They were in the trees. When they finally managed to trap some animals, they turned out to be black rats. Right? So normally rats live in buildings, they dig burrows underground, but somehow these rats have taken to eating pine cones and living up in the trees. That's weird for a population of rats. How did they manage to learn to strip these pine cones, right, to get at the seeds? Well, to do this, then you need to help with a biologist. So Eisner collaborated with biologist Joseph Turkel at Tel Aviv University. And they brought these pine-eating rats to the lab, right? So they have to trap some of these rats living in the trees, bring them to the lab, grab some city rats too of the same population, the typical rats who live on the ground and don't strip pine cones. Okay, it's the same species of animal, just two different populations. So now the starting point of thinking is that rats like what the foods they eat. They like what they can smell on their mother's breath. Okay, just like the rabbits I've told you about that learn in utero to like what their mothers have eaten. So maybe all you need is to have pine cones, pine seeds. All you need to do is feed pine seeds to the mother so that she gets the breath of pine seeds, and maybe the babies will like pine cones and then start stripping pines for the seeds. Okay, so they feed naive mothers, these non-pine stripping mothers with pine seeds, but their young fail to learn to strip the pine seeds, right? Well, maybe if they watch other rats strip pine cones, then these naive rats will learn by imitation. So they did an imitation experiment, and they basically gave up after 3 months. After 3 months of having a baby rat watch other rats strip pine cones, they still hadn't learned. This not that surprising after all, because rats are known for imitation. Okay, they're not very good imitators. At this point, I started thinking, well, maybe it's actually in the genes, right? Maybe it wasn't learned at all. It just unfolds with maturation. So then, to figure out whether it's culture, cultural transmission, or genetic inheritance, they had to do a cross-fostering experiment again, just like the one I mentioned last week. So they took some young rats from experienced, that is, pine stripping mothers, and from naive mothers, and they swap them around a bit, and you get this 2x2 design again. So young rats either had the genes of pine stripping mothers or not, or else they were raised by pine stripping mothers or not. And if genetics matters, then the right column here should be the ones that end up stripping the pine seeds. All right? No, that's not what happens. So the results showed, and couldn't be clearer, these results. You don't do statistics on these. It was the mothers who raised you that matter. If you're raised with a mother who can strip pine cones, then you end up learning, right? All of the young in this case. The genetic mother doesn't make any difference at all. Well, when they actually figured it out, well, here's the story, okay? And you make notes on the story because it's not on this slide. It turns out that the young rats are learning by a process, a shaping process that's unintentionally provided to them by their pine stripping mothers. So what you have to do is not chew this pine cone head on. You have to sort of peel back layers in this spiral fashion as is shown here. All right? But that's hard to come by if you're faced with a whole cone. But young rats figured this bit out, however, if the mothers let them have a pine cone that's almost completely stripped, okay? And so this basically happened because the mothers are letting their young ones steal the pine cones for them that they have been eating. So the young rats basically start with easy cases that have been almost all stripped, so they can learn to just finish off the job. All right. Then eventually, with the shaping process, they can do more. They can strip more and more of the pine cone, and most animals then end up learning this. Okay. So mothers have unintentionally provided shaping to the young rats because they let them steal a bit of food. All right. So because the young mums let them snatch food, they end up learning the skill. Well, this then leads to a whole way of life, including building nests in the trees. Right? So why not? All your food's coming from the trees. Why not start living in the trees? This saves traveling across the ground could be dangerous, right? So they've now carved out a whole ecological niche for themselves, right? A process called niche construction. They've constructed a whole new way of life, and it's passed down one generation from the other, right? Lord knows when it started, but it's been going for quite a while in Israel. Okay? We, with our cultural heritage, of course, and our cultural traditions, go for niche construction in a big way. So most of our environment is constructed, right, by our own devices. So most of you are probably listening to this now in some, constructed environment. That's what I would bet. I'm certainly coming to you from an environment that's been constructed, this Tinbergen studio. Skinner talked about thoughts and mental processes as covert behavior. Right? They hardly lead to any immediate consequences except through the behavior that they can engender. Now, however, with the advances in neuroscience, we can actually record from an animal's brain, and we can directly reinforce patterns of neuroactivity. Wow. So that's what this slide is about. And so electrodes were implanted in this case in an area of the rhesus monkey's brain called the parietal reach area. And this is roughly where the intentions to reach originate. The train of commands here goes from this intention area in the parietal cortex to the premotor cortex to the motor cortex, which of course then sends commands to the muscles to act. Okay, and you don't have to know this train of events here. Now, if you're interested, there's a little science article on this particular story. So what the experimenters do was first train monkeys to reach overtly with their hand to a spot that had been indicated on the screen. So crucially, this training required a waiting period. So a spot is first shown, right? And the spot then goes away, and the monkey has to remember the spot, right, in this delay period. And the monkey has to wait for a go signal before moving the hands or eyes. So no moving the eyes, no moving the hands ahead of the go signal, all right? All this time, presumably, it's got to remember what to do, right? And then think about doing it, but not to do it. So the monkey sort of have to think, well, where that green triangle is, right? Top right, got to move to top right, got to move top right, but naturally not move the hands and not move the eyes, okay? And when the signal goes on, the monkey then reaches over there and gets a reward, a little squirt of juice. The experimenter then tried to read the monkey's brain. Remember, it's got these electrodes in its parietal cortex. Now, while it was in this waiting period, presumably intending to reach and get the reinforcer, right? So then the experimenter tries to read the monkey's brain and reinforce the monkey for particular patterns of thinking. Okay, you get a moderate success out of this paradigm, mostly because the software can't really decipher the brain that well. So basically, monkeys on some kind of a partial reinforcement schedule, if you like, gets rewarded. Sometimes when the software figures out that it's intending to reach over. Okay. So what you can see, the difference between a think trial and a reach trial is basically the part that there's a component of the brain activity that has to do with actually ordering the hand to move. Okay, that's part missing on a think trial when the experiment tries to reinforce the monkey for just thinking about moving over there. All right. So when that paper came out in 2004, I wonder, well, what about human applications? And 2 years later, in 2006, a paper came out in Nature, documented a human paraplegic controlling the movement of a cursor, in this case, with brain activity alone. In this case, the electrodes were put into the primary motor cortex. And where's that again? Right. Okay. That's in the frontal lobe, right? And the motor cortex is on the top. So that's actually very easy to find it with an electrode. Okay, so patterns of electrical patterns of activity on the motor cortex then is used to control the movement of a cursor. And accomplishing that as a reinforcer for this particular subject, now he wanted to move a cursor around to read emails on the computer and do things like that. Okay, so using his brain activity alone, he's moving a cursor around. All right, you could see why this got into Nature. In 2008, a paper came out in Nature documenting the monkeys now moving a prosthetic arm to reach for food. So these are normal monkeys, okay? They don't have any brain conditions. And when they were participating in experiments, however, their arms were restrained. And electrodes were inserted again into the primary motor cortex of monkeys in this case. And they had to use the activity in the motor cortex to actually move a prosthetic arm, okay? An arm that's not there, just an electronic piece, to reach for a bit of food that's offered. And bring that food actually to the mouth, and that's a positive reinforcer for it. Okay, so the brain has to control this prosthetic arm enough to bring the food near the mouth so that they can get rewarded. Here's sort of what it looks like. So that arm there is not the monkey's arm, that's the prosthetic arm. The monkey's thinking its way to reach for a bit of food and brings it near his mouth and shoves it in. Okay, monkeys learn to behave a bit differently, so they're not nearly as coordinated with the prosthetic arm as they would with their own arms. So they actually move their face a lot. Their face actually moves toward the arm a good deal more. So a lot of the last bit is getting the mouth into the arm. This is actually the supplementary information from the journal article, so I downloaded it. Nowadays, articles often come with this kind of supplementary information. Boy, it's impressive to see this. Okay, let's turn to phobia treatments, right? Based on both these treatments that I'm going to tell you about. First one, systematic desensitization. Second one is flooding. These phobia treatments are based on models of phobia that treats the feared object as a conditioned stimulus. Okay, so in this case, you've got to remember that phobias are not random. We don't just get fears of any old things. In fact, some of the most dangerous things in this world, people hardly get phobias of. Cars, guns, knives, these artificial objects. Very few people are afraid to get into a car, for example, and yet it's a big killer in our life. Okay, the things we fear are non-random. There's things that in our evolutionary past have been dangerous, like spiders, snakes, for instance, right? High places. So, systematic desensitization is an attempt to replace the fear response that's produced by the stimulus with another response, a response that's incompatible with fear, and that's the response of relaxation. So let's take fear of snake as an example. Okay, first thing you got to do to do this therapy is learn to relax, and that's something you actually have to learn. It's not just kicking up your feet and opening a can of beer. That's not what relaxation's about. You have to actually learn to relax every muscle. So there's exercise that you can go through to try to do that. So part of it will involve try tensing one of your arms, right? Tense it as much as possible, and then poof, just let it go. All right. So you got to learn to tense and relax until every bone in your body, every muscle is actually relaxed. All right. So when you've learned to relax, then you make a list of hierarchy, right, of more and more fearful situations. You might start with just imagining the snake at a distance, right, to having a snake in front of you on a table, to finally holding a couple of snakes in your hands, right, in these gradual steps. So what you do is you learn to relax, and you then slowly go up the hierarchy in session after session. First you learn to relax while thinking of the snake, then you learn to relax while the snake's in front of you, then you learn to relax while you're holding the snake, and so on, all the way up. It takes a good number of sessions. It's got a good success rate, okay, but it takes a while because you've got to work gradually through the hierarchy, okay? You could call systematic desensitization the wussy way to get rid of a fear. Implosive therapy or flooding, I'm not going to distinguish though, is the macho way. It could work very quickly. What this does is an attempt to treat phobia by extinction. So extinction is the key principle here. In systematic desensitization is learning an incompatible response to fear. Here, it is extinction. What you do is you present the fear-provoking condition stimulus until the fear goes away. After all, if you're holding harmless snakes, nothing will happen, and fear will eventually goes away, right? It's very important in this kind of therapy to actually persist and not to stop with the first sign of fear, right? If you stop with the first sign of fear, that can have the opposite effect. That can actually sensitize the fear. So you got to fear till you fear no more. So let me illustrate a case. Hello, Mrs. Macquarie. You're here for your implosive therapy. You're scared of snakes, are you? Okay. Tell you what you should do. You should get in that cubicle there. Put on this hospital gown here. Change into that, right? Because in case the therapy scares the shit out of you. No, here, Mrs. Macquarie. Do it in the cubicle, please. Okay. Are you ready? Now what we're going to do is drop a whole lot of snakes on you, and boom! Okay, so all the snakes drop on the client, and client screams in fear until she finds out nothing happens and gets over the fear. That's sort of the idea of implosive therapy. Okay, you can get over the fear in a couple of sessions, rather quicker than systematic desensitization. Aversive therapy. Aversive therapy is based on positive punishment. Well, does positive punishment work? Skinner was famous for thinking, it doesn't work. He thought that it's sort of made for temporary suppression. Once the punishing contingency is removed, he thought the behavior would just return. But research, a lot of subsequent research has shown that punishment can work. But to work, it needs to have these principles, right? The positive punishment. It needs to be immediate. Okay, it's no good waiting till dad comes home, right, or until the principal arrives in the afternoon. If you're going to use positive punishment, it's got to be immediately after the behavior. It's got to be consistent, right? There's got to be no wriggle room getting out of the punishment. So every time the behavior is emitted, the punishment has to be given with no escape possible. And it should start at full strength. It's got to start at a strength that's strong enough to actually have an effect. It's no good trying to ramp up with small punishments until you ramp it up to somewhere big. Okay, so there's studies actually doing that with shock levels in rats. So if you ramp up a shock level slowly, then you actually have to arrive at a far higher shock level before you have any punishing effect than if you start at, say, some medium strength shock. Okay, that's the result we find with rats. So if you ramp up, you got to ramp up the punishment a lot before it works. And finally, it should not be a cue for positive punishment. So punishing a child should not lead immediately afterwards to hugs and kisses. Okay, so we don't want the punishment to be a cue for positive reinforcement. It helps very much, right, if you're trying to reduce a particular behavior to have another behavior that the animal or kid can engage in to get whatever it was that the behavior brings, right? So if you want children to be quiet in the house, then it helps if they're allowed to go outside and make a lot of noise. So they have an alternative for which they will not be punishment, and they'll get they'll get whatever rewards they get out of yelling and screaming. Okay. So positive punishment is useful for some desperate measures, right? And electric shock can be used as a punisher, even with humans as well. I'll tell you about a couple of cases. So some authors along the way have used these shock therapies, made up this rather facetious term, faradic therapy. It means giving shocks to kids, okay? God knows, psychologists can be so bleh. Anyway, so good at making up terms. So in one case, well, in a number of cases actually that have been published, you got these babies that are desperately thin and starving because they've been constantly vomiting, all right? So you feed them some food and then they'll vomit it up. And this leads to a very skinny baby, as in this before picture. And in this case, the shock therapy was applied as a last-ditch measure. There's nothing else that has worked, and this baby is in danger of dying of starvation, right? At least it's suffering malnutrition. So the idea here was to use shock therapy to try to stop this vomiting. So the idea is to shock the baby on the leg at the first sign of vomiting, okay, after feeding. So note that this is a contingent application of shock. You don't just shock the baby randomly. You shock the baby every time it starts vomiting. Okay, that makes it far less aversive in this case, because the baby can do something about it. The baby can stop vomiting and stop the shock. Okay, so that's what all these funny contraptions about. It turns out in later research, so that's an attempt to measure just the first muscle movement of vomiting. It turns out that's overdone. And later research showed that's not necessary. You can just punish the baby the first time you see some spew coming out. Okay. So, cut a long story short, it worked. After a few sessions, vomiting actually stopped. Okay. They stopped the sessions, the baby started vomiting again, and a couple more sessions then handled this spontaneous recovery and got rid of it. And the end is, at the end you get a baby who seems to be healthy and become far more sociable. Okay, so at least we don't know what side effects it might have had, this kind of therapy, although we could, and it's hard to separate any side effects from the therapy versus the any effects from the before conditions, from all the starvation. Okay, but at least no adverse side effects were detected. The baby actually became more sociable, right? This kind of therapy using electric shock has also been used on kids who are self-destructive. So some autistic children are hell-bent on self-destruction. They bite themselves, or they like to bang their heads very hard into the wall, and they can do serious head damage and serious brain damage. You could reduce this kind of behavior, pretty much stop it, or at least reduce it a great deal when such behavior is contingent on shock. Okay, so every time the kid runs into the wall, you shock the kid. Don't know why it works. Kids who are willing to take the pain of slamming their heads into the wall will stop that when given contingent shock. You can go figure, but it does work. This kind of therapy has been used as a desperate measure to stop kids from engaging in such self-destructive behavior. The practice of shocking kids, of course, remains very controversial. Right? Even if it works pretty well, you could think of the ethical issues involved here in using serious punishment of this kind to try to stop unwanted behavior. Let me summarize. We looked at evidence for episodic-like memory in Western scrub jays in a task that required the birds to combine information on what, where, and when. They need to combine what food they had stored with where they have stored, with information on when they stored it in order to do the task. These three elements together, what, where, and when, make up episodic-like memory. We looked at the case of cultural transmission in black rats. This is made possible by social interaction between young rats and their mothers. It's not imitation, this social learning, this socially mediated learning, but it's a kind of socially facilitated operant conditioning, if you like, or inadvertent shaping procedure on the part of the mother. Young rats learn by finishing off the pine stripping job that their mothers had started on some cones. And they get to do this because the mothers are allowing them to steal the food they were eating. We end up with cultural transmission of a whole way of life in this population of rats by purely behavioral means of transmission, this socially mediated operant conditioning. We then looked at three cases of applications using learning principles. We looked at positive reinforcement of cortical activity, either read directly or used to move a cursor or move a prosthetic arm. Okay, so in one case, the experimenter handed the monkey rewards for directly on reading the brain activity. In the other two cases, the brain actually operated devices, okay, a cursor or prosthetic arm. We looked at two treatments for phobia, systematic desensitization, which is based on learning an incompatible response of relaxation, and flooding, or implosive therapy, which is based on the principle of extinction. We then looked at using contingent shock as a positive punishment in therapy, and this is used as a desperate measure to stop catastrophically harmful behaviors such as vomiting in babies and self-destructive behaviors like headbanging in children.